Hi y'all. As promised, I'm going to upload a quick overview on chapter 9, the cell cycle and mitosis. So technically, you should have been done with this module yesterday, but for those of you who are a little bit late, for those of you who have not been signing into virtual office hours, here is a quick overview on chapter 9, the cell cycle, and what we need to keep in mind. So what we are talking about, I'm actually going to scroll to this slide really quick because we are talking about the cell cycle as a whole. So we have multiple phases in the cell cycle, and we want to understand kind of how a cell goes about living its life. And depending on what type of cell you are, you're going to have a different life cycle. For example, perfect example of this is if we're talking about eukaryotic cells, right? We are eukaryotes, we are multicellular, we are mammals, and we have many different cell types in our bodies that undergo the cell cycle at different rates, and some actually don't go through the cell cycle at all. So if you look at the slide right here, the eukaryotic cell cycle, we have two broad phases of the cell cycle. The cell is either in interphase or it's in the mitotic phase. And the first part of this chapter talks about the mitotic phase, the M phase, mitosis, and cytokinesis. And that's how we make new cells. Okay, if you are in interphase, this is kind of where the cell is just doing its thing, living life, and specifically G1. So we can either be in interphase or we can be undergoing cell division. In order to get ready to undergo cell division, there are a few different things that the cell needs to do to prepare, and those are the S and G2 phases. And we'll go into a little bit more detail in that. I just wanted to let you guys know what we mean when we say the cell cycle, okay? So first we're gonna talk about mitosis. I posted these slides. I made a couple of little adjustments here in this video. I deleted one slide and changed one key concept, but these are the key concepts that I want you to understand for the purpose of this class and this lesson, okay? So that the first thing is, living cells are going to undergo the cell cycle. They're going to go through the cell cycle at different rates, okay? If it is not going through the cell cycle, a cell is going to chill out in a phase called G0, and we'll talk about that, okay? We're also going to talk about the structure of DNA and chromosomes. You have your vocab words here. I know a lot of them sound very similar and confusing. Chromosome, chromatin, chromatid, sister chromatid. We will go through that, okay? The events that take place in mitosis, you have to know the events, okay? And your book does have an extra phase in there than the videos you watched, and we'll talk about that as well, okay? And then the checkpoints that the cell has to go through in order to progress through the cell cycle, because if you are progressing through the cell cycle uncontrollably, cell cycle gone wild, that is the disease of cancer, right? And there are so many different types of cancer. We still do not yet have a cure. We have treatments, but it is a disease where the cell is just going through the cell cycle uncontrolled, cell cycle gone wild. So we will talk about one example or one type of process where cells do not heed the checkpoints for the cell cycle. Okay, so I'm gonna go into presentation view. See, I'm just figuring out this online stuff. Okay, here we go. All right, so first of all, your worksheet and some of the stuff you're working on, questions, you may see this question: why do we undergo mitosis? Well, there are unicellular organisms like bacteria, right, or unicellular eukaryotes that undergo cell division they replicate themselves to produce a completely new organism. So reproduction is one important thing specifically for single-celled organisms. And let's remember that in these next few chapters, we're going to be talking about genetics, and we're going to be talking about, for example, the next module is going to talk about meiosis and sexual life cycles. We're going to focus on the human genome, human chromosomes, eukaryotes. Always keep in mind that different organisms do this differently. For example, your textbook talks about binary fission, how bacteria divide and replicate, right? So slight variations, similar mechanisms. So just remember that we are going to be focusing on eukaryotes here, okay? But there are single-celled eukaryotes like protists that divide to make new cells, okay? So reproduction is one thing. Growth and development. If you watch the video from Mr. Anderson, he had a little picture of a teeny tiny human embryo, right? The way you come to be is mitosis, right? You go from this little tiny, first you go from a single cell, right? A fertilized egg, which is a zygote, and then you undergo a whole bunch of mitosis and cell differentiation to become you. So growth and development need mitosis for that. And then if you get a boo-boo, right? If you are riding your skateboard or running along a trail or something and you get a boo-boo, a cut, a scrape, we've all had that happen, even something as simple as a paper cut, right? You need mitosis in order to get tissue renewal, in order to repair your tissue, as well as everyday wear and tear, right? So we're going to talk about the difference, for example, of epithelial tissue, your skin, versus other types of tissue, right? We shed our dry skin, we exfoliate our skin, we get rid of lots and lots of skin cells. Skin cells are highly mitotic, right? And hopefully it's regulated, because if it's unregulated, you get a lump or a tumor, right? So tissue renewal is another reason. So those are the three reasons why we undergo mitosis. And then when we undergo mitosis, when a cell undergoes mitosis, really, really important thing that happens is creating two genetically identical cells. Okay, so now that I've said genetically identical, we have to kind of talk about DNA and what we mean when we talk about the different structures and different conformations of DNA. So this semester, y'all have already heard about the central dogma, right? That is when we go from DNA to RNA, we do transcription, and then we do translation to make protein. Right? So thus far, you've heard about DNA, you've heard about the double helix, sugar phosphate backbone, nitrogenous bases, apples in the trees, cars in the garage, right? You've heard about DNA, deoxyribonucleic acid. You know that humans have 46 chromosomes, right? You have 46 chromosomes in all of your cells except for eggs if you are female, biologically female, and sperm if you are biologically male, right? Those cells only have half of the amount of chromosomes. They have 23 chromosomes. So this figure is showing you a chromosome. Okay, the middle part of the chromosome is called the centromere, and this just so happens to be a duplicate chromosome. Okay, it's still a chromosome, it's a duplicated chromosome, and we'll talk about where that happens. This term right here, chromatin, when we say chromatin, we are talking about DNA and the proteins that it associates with. Okay, because we have a really, really, really long structure, right? Typical human chromosome is going to be something like 2 meters long, and we have to be able to squeeze that or tightly fit that in along with 45 other chromosomes into a nucleus, right? So the way that we package DNA is we wrap it around proteins, and there are a few videos. This chapter doesn't go so much in detail about that. We learn about that in chapter 14, but on your online module, you have hopefully watched that animation that shows you how DNA is packaged, right? And it wraps around these proteins called histones, and that's what we mean when we say chromatin, and then it has several other structures, right? So when we are talking about this central dogma right here, right? Gene expression going from DNA to RNA to protein, this is happening during interphase. It is not happening in mitosis. In mitosis, our one goal is to divide and make two genetically identical daughter cells, okay? The karyotype, a karyotype is just a way to visually represent 46 chromosomes in the human genome, okay? So you know that you are half mom, half dad, right? Maybe you got your mom's eyes, dad's nose, I have my dad's nose, and then you have some behaviors that maybe you do similar things as mom, similar things as dad. It's because you're half-half, right? You are this beautiful mosaic, half from mom, half from dad, and that is actually reflected in your genetic makeup. So what a karyotype is a visual representation of actual human chromosomes, right? In this case right here. So this scientist right here is doing some really cool fluorescent microscopy, visualizing chromosomes, and then how you get this karyotype. If you Google a karyotype, or if you have watched the videos in the online module, they're all lined up really nicely in pairs from 1 to 23. Right? That's not how they look in your nuclei, right? What happens here is you have a really cool scientist doing fluorescent imaging, and then they use some kind of imaging software like ImageJ or whatever kind of software that particular lab is using, and they organize it in this karyotype, okay? The pairs, one from mom, one from dad, have a name, right? They're called homologous chromosomes because they are similar chromosomes. And if you look here, right, this color coding right here in this homologous pair, like you got like this pink tip right here, it's because they express or they encode for the same genes, but you got one from mom, one from dad. So they're not the exact same, but they are similar, and they have genes in similar locations, also called loci is plural or locus is one. And we will learn more about that. So those are your homologous pairs. Humans are diploid or diploid, right? We are 2N. Okay, and we're going to talk about haploid cells when we talk about meiosis, okay? So when a chromosome has been duplicated, right? You have, let's say this is your one, pair one, and you have, this is one chromosome and this is a second chromosome, right? These chromosomes are duplicated. And you should already know by now that they were duplicated in which phase of the cell cycle? Synthesis, right? The S phase. So they are duplicated chromosome. This is an exact copy of this, but they are attached to each other right here at the centromere, okay? The centromere. Overview of mitosis. So what happens? Goal of mitosis. Think about, you get a boo-boo, you're going to make skin cells that were exactly the same as the skin cells surrounding them, that were exactly the same as the skin cells that got scraped off when you got your boo-boo, right? So the goal is genetically identical, we call them daughter cells. Okay, so if you look here, okay, we start with a cell where this is just representing one cell, right? But keep in mind, you have 46. It's a lot more confusing. If you watch all of the videos on mitosis, we cut down the number of chromosomes just to make it simpler, right? So you have one chromosome there. It is not duplicated, right? It's just a regular old chromosome. And then when the cell is ready to undergo mitosis, you duplicate the chromosome, and now it has sister chromatids, okay? It's still one chromosome. It is a duplicated chromosome. It has sister chromatids. And then once you divide your cell, you separate the sister chromatids, and now you have two cells that are identically the same, genetically identical, okay? All right, cell cycle. Let's take a closer look. So we have two parts, interphase, okay? Gene expression and DNA replication take place there. So that is when the cell is living its life, doing its thing. We learned in chapter 5, someone had a really good question in the virtual office hours about like, when does membrane transport happen, right? Like we talked about, for example, passive transport and oxygen and carbon dioxide diffusing through the lipid bilayer, right? That's always happening, right? That's not going to stop happening, otherwise your cell would die. But maybe like responding to external signals and things like that, responding to, if you had like a signaling molecule binding to a receptor and you had signal transduction, reception, transduction, and a cell's response, right? That's happening right here in G1. That's cell growth, okay? So interphase is the cell living its life, doing its thing, specifically G1, and then it gets that growth signal, all right? Then it gets that growth signal. I go out into the hallway, I open the door, and I say grow, and I want you to transduce that signal, right? What I mean, or what the signal mean, the growth factor, right? That signal could be a growth factor. It's telling the cell to grow and divide. So the cell then goes into S phase, and this is where you go from a chromosome to a duplicate chromosome. All right, so you double your genetic information, but you still have 46 chromosomes. They are just duplicated chromosomes. Okay, that is where DNA is replicated, S synthesis. Okay, then now the cell is getting ready to divide. It's doubled its DNA, but guess what? It needs to double everything else, right? Not necessarily double, but it needs to replicate everything else, all of the organelles, the ribosomes, the amount of phospholipids that it has there to make the ER, to make the Golgi, to make your lysosomes, to make every single organelle that we talked about in chapter 4. Now you need enough organelles for 2 cells, because you're eventually going to divide into 2 cells, okay? And that's what happens in G2. Once you do that, then the cell goes into M phase or mitotic phase, and it's going to go through several processes in order to become 2 new cells, okay? So duration of a life cycle, duration of a cell cycle, not the life cycle, is going to vary cell to cell, right? Depending on what type of cell you are. For example, I'm sure you guys are all aware of this concept that you've heard this before, that neurons in your brain do not undergo mitosis, right? Muscle cells, they are highly amitotic, okay? They're actually called terminally differentiated in the literature, right? So they're not going through the cell cycle. They're still responding to signals. They are still doing their thing. They're doing the central dogma. They're doing transcription and translation and maybe movement and signaling and active transport and passive transport, right? But they have no intention of going into mitosis, and the phase they're chilling in is called G0, okay? G0. All right, so if we want to go through a little bit more detail on what happens during mitosis, let's first, so the acronym you've seen so far is PMAT. Okay, I'm gonna go with what your textbook talks about. It's not contradictory. Your textbook is just looking at mitosis in slightly more resolution, a little bit higher resolution, in that we have this extra pro-metaphase right here. And it's just looking at it in a little bit more detail. Okay, so interphase, we have not entered mitosis yet. G1, the cell is growing, doing its thing. In S, it is doing DNA synthesis. And then in G2, it is replicating the organelles. Okay, so in G2 of interphase, we know the cell is going to go into mitosis. It's getting ready to go into mitosis. It's going to replicate these structures here called centrosomes. And the centrosomes are what produce those spindle fibers that help to pull apart the chromosomes during mitosis. Okay, you can think about that spindle like if you were trying to move something into a building, right? And you want to do it in a very strategic way. So you get a pulley system with chains and you attach the chains. Like let's say you have like this really fancy piano, right? And you want to get it into a building, right? You're going to do it with a very structured system, right? So the spindle is the way that we take the chromosomes, attach them to these fibers, and pull them into two new nuclei. Okay, so centrosome is going to replicate in G2, and then there's this concept you have to understand when DNA is condensed and when it is not condensed. Okay, when the cell is doing its thing, central dogma, gene expression, right? You want the DNA to be accessible and you want it to be able to interact with transcription factors and things that control gene expression. So it looks like this, right? This is a stylized cartoon, but the point here is that it is uncondensed. You don't see distinct chromosomes. Okay, and that is an interphase, right? So then once you go into mitosis, the first phase of mitosis is called prophase, pro meaning before, right? This is where chromosomes begin to condense. They want to get really, really, really organized and condensed. Okay, you got your sister chromatids. They were duplicated already. When? Which phase? All right. S phase, right? They were already duplicated in S phase. Now they condense, your nucleolus disappears, and the spindle begins to form. So you start to make these nice long microtubule fibers so that you can use them to pull apart your chromosomes. Okay, so prophase, you see condensed chromosomes and you see an early mitotic spindle. Then our next phase is prometaphase, and you don't see this in a lot of the videos. It's just looking at this process in more detail, right? In prometaphase, this is when we see the nuclear envelope or the nuclear membrane begin to break down, okay? Because we talked about the nuclear envelope, it is isolating the DNA, it's keeping it packaged in a very, very, very safe space. Now that you're ready to divide into two new cells, you want to fragment that nuclear envelope and make them accessible to these microtubule spindle fibers, okay? So in prometaphase, nuclear membrane fragments, or you don't see a distinct nucleus anymore, and your microtubules, your microtubules start to attach to the chromosomes, and they attach to the chromosome at a very specific part right over here. What is that called? It's called the centromere, right? That is where microtubules attach. Your book gives you the specific name for the microtubule when it is attached to the centromere, okay? That's this one right here, sorry. Give me my pen. Okay, that's this one right here. The kinetochore microtubule. Help. Okay, the kinetochore microtubule. There we go. Kinetochore microtubule is what attaches to the centromere, and you have one attached on one side and then eventually one attached on the other side. Okay, so in prometaphase, no distinct nucleus, you don't see a nucleolus anymore, and you have your microtubules start to attach to the centromeres of each chromosome. Okay, then we go into metaphase. Meta, middle. Look at how different this looks, right? This is an easy one. This is the one that you should be able to always identify, whether we're talking about mitosis or meiosis, right? So in metaphase, you have all of your chromosomes lined up at what's called the metaphase plate. In the middle, one sister chromatid is attached to one side, right? And the other sister chromatid is attached at the other side, right? So you have one microtubule attached here and one microtubule attached here to the same chromosome, right? And the way they get there is they push, pull, push, pull, push, pull until everybody is lined up at the metaphase plate. This is a really, really important place in mitosis. There is no turning back once you separate them. So if you separate them incorrectly, you are in big trouble, right? Because then you end up with two cells that are not genetically identical. Okay, so metaphase plate is an important checkpoint. Okay, so metaphase is an easy one. Everybody lines up. Okay, and then anaphase, ana or apart, right? They get separated. Now look what you got. In metaphase, you had, how many chromosomes do you have there if that was a human cell? 46, right? You have 46 duplicate chromosomes. Here, you've taken your duplicate chromosomes and you've pulled them apart. And you're not quite in two cells yet, right? So if there was like a trick question there, right? Now you have double the chromosomes. So now you have 92 separate chromosomes there. Sister chromatids are pulled apart and now you have separate chromosomes that are no longer duplicate chromosomes, okay? There are these proteins once upon a time that held the sister chromatids together at the centromere, right? You have, let me scribble, go away. Right, that's your centromere. Here's your X, sorry, and there's your centromere. Within the centromere, you have these proteins. They're cohesive proteins, right? They're called cohesins. They hold the sister chromatids together, okay? In order to separate them, right, they're attached to kinetochore microtubules. In order to separate them, you need to degrade those proteins. Okay, so that's what that means there. And then we get into telophase. Okay, now in telophase, you start reforming your nuclear envelope or your nuclear membrane, and you start to see two distinct nuclei. You get your nucleolus back, your spindle fibers disappear or disassemble, and we have cytokinesis. So mitosis, we've created our two, we want to create our two genetically identical daughter cells. Cytokinesis is the process by which you separate the cytoplasm. Cytokinesis, movement of the cell, right? So telophase and cytokinesis are usually grouped together. Cytokinesis is specifically talking about separating the cytoplasm and pinching off into two cells. And there's a really cool structure associated with that. It's called the cleavage furrow. Kind of looks like a butt, all right? So in an animal cell, it's gonna happen differently than in a plant cell, okay? So there is this contractile ring of cytoskeletal proteins, okay? Actin and myosin come together and they kind of pinch off the plasma membrane so that now you have two identical daughter cells. So when you see cytokinesis, you should think cyto means cell, something to do with the cell. Kinesis is movement, right? So now you go from one big globule with two nuclei to two distinct cells. In plant cells have this specialized structure, right, that make them different from animal cells or fungal cells or whatever. They have a cell wall, right? And in order to undergo cytokinesis, you have to basically make a new cell wall right here in between, right? So what happens is you get these vesicles full of the materials you need to make the cell wall in plant cells, and you form what's called a cell plate. And now you have a new cell wall, and in the process of making that new cell wall, you see right here, you have these vesicles. So they actually get sent from the Golgi to this area right here, and then you are able to pinch off the plasma membrane with that new cell wall. Okay, so when you see cell plate, you are actually making a plate between the two cytoplasms so you can create two genetically identical daughter cells. Okay, so that is mitosis. Okay, remember your, that is terrible. Sorry, mat. Okay, prophase, we're starting, the DNA is condensing. Prometaphase, you lose your nuclear envelope, you start to see your centrosomes, your spindle fiber, your early mitotic spindle forms. Metaphase, middle. Everybody is lined up at the middle, right, at the metaphase plate. Anaphase is apart. Telophase, you start to form your two new nuclei. Telophase goes together with cytokinesis, and you pinch off into two separate cells. Okay, so I hope that made everything a little bit more clear. And now we just want to talk about cell cycle gone wild. What happens when the cell does not heed the checkpoints, number one? And what are those checkpoints? How does the cell make sure that it's progressing through the cell cycle properly? What are the signals it's looking for, right? So there are 2 points where that cell cycle is irreversible. Once you've replicated your DNA, once you have duplicate chromosomes, you can't reverse that. And then once you've separated the sister chromatids, you can't reverse that, right? So that means that you really need good checkpoints to make sure that you should do that in the first place, okay? And there are so many proteins and enzymes associated with those cell cycle checkpoints that we do not talk about in this course, okay? You can learn all about them in cell biology, okay? So what I want you to know is that there are 3 checkpoints and what each one does, okay? So this right here is what phase of the cell cycle is that? That is interphase, right? You're either in interphase or you're in the M phase, okay? So in S phase, what happens? That's where you duplicate your DNA. So before you duplicate your DNA, being that that is an irreversible process, you want to make sure that you are responding to the appropriate signals, okay? It's called the G1 checkpoint. You are responding to a growth factor, right? Like an example of a growth factor that is associated with a particular type of cancer, growth factor receptor, is called EGFR. It's epithelial growth factor receptor. And there is a mutation in this growth factor. I know I keep touching my face. I'm not supposed to, but I just washed my hands, so I'm good. EGFR is this is just, you don't have to know this for the exam. I'm just giving you an example of a growth factor where you have a mutation in the receptor and it is associated with particular types of cancers like lung cancer. Lung cancer is one of them, right? So when you see something called growth factor, it's an actual signaling molecule that binds to a receptor that tells the cell, grow. Okay, so the G1 checkpoint is going to make sure, with a whole bunch of proteins and enzymes and stuff, that the cell is indeed responding to the appropriate signal. The second thing it's going to check for, which we'll look at, it's the last part of this chapter, do I have space to grow, right? Like if you get a cut and you repair that cut on your hand, there is a signal with your cells are talking to each other and letting each other know that, hey, we've covered the space we need to cover. We don't need to grow anymore. Let's stop growing because there's no more space. Okay, so that's what the G1 checkpoint does. G2 checkpoint is right here in the cell cycle. I want you guys to always kind of envision this diagram. It really helps you keep track of where you are and what happened. Okay, at the G2 checkpoint, look what you've done already. Okay, so you passed your G1 checkpoint, you went into S phase, and then you went into G2 phase. What is the thing that you did that is irreversible? You've duplicated your chromosomes, right? So before you go into M, before you go into cell division, you want to make sure, did I duplicate my chromosomes correctly, right? So the G2 checkpoint assesses DNA replication, okay? And then it's gonna say, okay, good to go to mitosis. Now we are committed to cell division. Okay, so we go through cell division, we go through prophase, prometaphase, metaphase. That's important, right? Metaphase. You want to make sure that everybody, before you pull them apart, you want to make sure that all of your chromosomes are lined up at the metaphase plate properly, right? Let's say this is my duplicate chromosome. I want to make sure that this chromosome is attached to a spindle fiber here, and it's attached to a spindle fiber here so that they get separated into the two new daughter cells. Okay, so the M checkpoint is making sure that all chromosomes are attached properly to the spindle at the metaphase plate, and then whoop-whoosh, then you get two new cells, and then guess what? Life goes on and you go back into G1, doing all of the adult functions of the cell, responding to signals, active transport, passive transport, sodium, potassium, ATPase, all that stuff is happening, right? It's also happening in mitosis, but not all of the things, right? Like something like, let's think about what we learned about in chapter 5, like what would not happen during mitosis in terms of cell signaling and a cell responding to its environment. What can you think of? Like oxygen, carbon dioxide exchange, that has to happen, right? Sodium, potassium, that has to happen. You have to maintain good osmotic pressure. You don't want your cells to shrivel or expand or all these processes, right? What about something like a steroid signaling molecule? Where do you find the receptor to a steroid signaling molecule? Review. In the cytoplasm, right? Because typically like something like a steroid hormone is going to go into the nucleus. Once it binds to its receptor in the cytoplasm, it's going to go into the nucleus and do gene transcription, right? If you are busy making two new cells, you are not doing gene transcription, right? Your DNA is compacted very tightly into visible chromosomes. It is not available for gene transcription. So gene transcription is not happening during mitosis. Whereas now that you go back into G1, you kind of unpack your DNA, and then you get back to living life. Good analogy that we talked about in the virtual office hours is like moving house, right? If you're going to move from one place to another, you're going to pack all of your belongings, which I will be doing at the end of this month. I will be packing all of my belongings into nice, organized Tupperware bins. I will label them. I will know where everything is, but can I go about my day as I do right now when all of my stuff is in a certain place? No, I can't. I have to wait until I get to the new place. I got to unpack all of my stuff, uncondense it, put it all over the house so that I can go about my day and teach and live life and do my thing, All right, so that's a good analogy for DNA packaging. When it's packaged tightly into a chromosome, you ain't doing no gene transcription. Okay? All right, so that's just a repeat of the checkpoints. Okay, and let's just quickly look at what happens when cell cycle goes wild. Okay, one of my friends actually taught an entire class on cell cycles gone wild, cells gone wild. Okay, cancer, this is what happens. So the experiment in your book, and you can do this in a lab if you ever get an opportunity to do some cell culture in a lab. It's really cool. You can grow a particular cell type in a dish with the right nutrients, right? And what you will see is that they will grow, if they are normal healthy cells, they will grow like this until you get like this nice cobblestone shaped structure, and they're touching the whole ends of the dish and they're all touching each other, and then they stop growing. Okay, this is called density dependent inhibition. The cells are not going to progress past the G1 checkpoint. They're not going to bother duplicating their DNA. They're not going to bother duplicating all of their organelles because they have reached the amount of space that they have for growth. Okay, cancer cells, they don't follow the rules. They blow past the checkpoints because something is wrong there, right? There could be so many things wrong there because they will grow even when they don't have the appropriate growth factors, right? So they'll grow and make these clumps, right? And if they don't have density dependent inhibition, and once you get this growth, too many cells in one place, that's a tumor, right? It could be benign or malignant. If it's benign, it's not really going anywhere. Like maybe you've had a dog with a benign tumor. Doggy has like a little fatty tumor on his belly right now, right? It's not dangerous, and we just keep an eye on it and it's not growing. If it starts to grow, or a piece of it goes off and goes and flows somewhere else in the body, that's called metastasis, and that is a malignant tumor. Okay, so really important to understand the whole big picture of the cell cycle. What's happening? You have two parts, interphase and mitosis. The goal of mitosis is two genetically identical daughter cells. 3 reasons why we do mitosis, right? Keep this in mind because the next lesson is about meiosis, and they sound similar and they look similar. Okay, I will post the next video ASAP. Good luck.